Well, what's up, Sci-Fam? It's your boy Nickel here, and we're gonna go magic school bus style into the nucleus. Remember, the nucleus is found in a cell. So, as we talk about cell division here in mitosis, it's kind of important that we consider about where we're at in this whole process. So, go ahead and title your page into the nucleus. You can see we're going to define nucleus. There is a paper you need to get out of the extras bin, a little square that we'll use as a diagram to kind of look at how DNA is somehow folded and packaged inside of the cell. So go get that out of the extras bin. And we're back. Make sure that you just see how we're going to lay out our page. So tape it in when we get to that part in the video. So let's jump into the nucleus. So let's just define nucleus. Or at least when you research what a nucleus does, a lot of times you'll see that it's called the control center of the cell, because the DNA is inside of the nucleus, and the DNA is basically instruction manual on how to do its job, how the cell needs to do its job. So in the nucleus, it's going to, the nucleus itself is going to hold DNA in a secure and safe location, because if DNA gets damaged at all, it'd be like removing 1 or 2 or 3 or 5 or 100 words out of an instruction manual. You can't accomplish the task as easy if you don't have all the instructions. So we want it to be secure. So so the cell has a nucleus which holds that DNA secure and safe, and the nucleus is going to surround the DNA, kind of like with a city wall, but not with a city wall, but with a membrane. Kind of like the outside of the cell. It's like it creates a pouch so the DNA can live inside of the nucleus. So we just don't call it a membrane, we call it the nuclear membrane because it's related to the nucleus. So it surrounds the DNA with a nuclear membrane. So if we now tape in this diagram here, this kind of orange wall here is the nuclear membrane which surrounds the DNA. And we have like multiple feet of DNA inside of every cell, which is just mind-blowing to think about. Well, how we actually fit it in is we have these little tiny circular proteins called histone proteins that form, and if we zoom in there, you can kind of see them a little bit better. They form and they start to wrap up our really, really thin strands of DNA, and as they wrap them up more and more, they start to create this form here. So this stuff with these histone proteins grabbing our DNA, we call this chromatin. So chromatin is basically the yarn that makes up a chromosome. So if we were to kind of like relate this, chromatin is like yarn and chromosome is like a sweater. A sweater is made out of yarn. The yarn wasn't all wrapped up and organized, but when it is all wrapped up and organized, you can make something like a sweater. Well, DNA, when it's not wrapped up and organized, it's called chromatin, so that'd be like the yarn. When we then wrap it up and organize it, we can then form something that we call a chromosome. And so as we go through the process of mitosis, it'll be important to know the difference between chromatin and a chromosome, and how both of them are actually just DNA, but based on how the DNA is structured and wrapped is going to change its name. So yes, a sweater might be made out of wool, but we wouldn't call it a wool, we would call it a sweater because it has a new shape. Yarn is made out of wool, but it has a different shape than a sweater, so we give it a different name. We call it yarn. Well, chromatin is made out of DNA, and it's like the yarn. Chromosome is made out of DNA, and it's like the sweater. They're different products, so we have different names for them. All right, so we're going to make sure you label and annotate your diagram just as I did mine, and label the nuclear membrane, label where it looks like chromatin, and then circle the chromosome to show that this kind of X-looking thing is what a chromosome is, and that all of them are made out of DNA. So to summarize, DNA in a cell. So DNA in a cell's nucleus. Nucleus is usually, so like 80 to 90% of the time, in the form of that stuff that we call chromatin. because it's easier to be read when it's in chromatin. When it's all packaged up, it's like it's all stored away in filing cabinets and stuff, and that's when it would be in the chromosome shape. So it's hard to read it if it's all stored away. So when it's not all stored away, it's a little bit unraveled. It's in that chromatin, and 80 to 90% of the time, if you were to look inside of a cell, 80 to 90% of the time, you'd find the DNA in the form of chromatin. And remember, chromatin is just slightly condensed DNA. When it's even more condensed, it turns into a chromosome. So chromatins make chromosomes. And there you have it. All right, now that we've kind of looked at the inside of a cell and gone into the nucleus, we can now use these new vocabulary terms when we talk about DNA as we learn about how DNA is copied and separated into two identical cells in the process of mitosis. I'll see ya over there. Let's go.